DNA replication is the copying of DNA, and it is necessary to replicate the DNA before cell division so that the two daughter cells have the same DNA. And after they have discovered the structure of the DNA, the scientists proposed 3 theoretically possible methods how this DNA can be copied, and they're here on the left side, namely the semi-conservative replication, conservative, and dispersive replication. Of all of these 3, only the first one here, the semi-conservative one, is actually the correct one. And that's the one that you certainly have to know. The other 2 down here, the conservative and the dispersive replication, these were theoretical possibilities, but the scientists did not know which one of these is correct. So in that sense, it's interesting to also know a little bit about these 2, because they used an experiment to exclude this possibility down here with the conservative and the dispersive. So, but now let's have a look. Over here, the red double helix over here, that's our original DNA. And this original DNA we also label as template DNA. So that's our template, our DNA template. And here what we have right now is this DNA is copied, and then we have 2 DNA molecules. And each DNA molecule contains, in this case the red one, this over here, the red one over here, that's the original one. And the light gray one, that's the new one. That's the new DNA. And the same down here, we have over here an original one and a new one. And because the 2 DNA copies now each contain the original DNA, and because the original DNA over here is conserved, we refer to this as the semi-conservative DNA replication, because the original DNA is half conserved in the 2 new copies. In the case of the conservative DNA replication, which does not occur in nature, we keep the original DNA and we have a completely new DNA down here. But as I already mentioned, this was basically, that's what the scientists considered as maybe as a possibility, but essentially does not occur in nature. And in dispersive, you can actually see that the 2 DNA copies contain over here original, over here new, then over here again original, and down here we have new or original DNA and new. But also this one does not occur in nature. So that is basically the general overview. So from now on, we're only going to be concerned about the so-called semi-conservative DNA replication. And I'm going to explain you now how this happens in more detail. So over here you see a DNA molecule. I have to change now my pen color again. That was not the correct one. So here's the pen color. I turn it to white again. So over here I have basically the original DNA, and there are 2 enzymes necessary that you need to know. And the first enzyme is name is helicase, and you know it's an enzyme because of the ending ase here at the end. And this enzyme helicase is responsible for unzipping or also as unwinding DNA. This enzyme can be found over here, right where the 2 DNA strands separate. So that's basically our helicase enzyme. I'm going to later remove it again so to make it easier to see. But right now, and this helicase moves into this direction over here and separates this strand of the DNA and this strand. And you see down here that they're already separated. And that's the job of the helicase. In this process, it breaks hydrogen bonds. And these hydrogen bonds can be found here in between the bases, between the G and the C and the A and the T. This is basically where you find the hydrogen bonds, and the helicase breaks these hydrogen bonds and separates it. And in order to, that's the next one, DNA polymerase. But before I talk about that, I'm going to remove all these lines again to make it a little bit more easy to see. Okay, and DNA polymerase, that is now the second enzyme which is necessary. The DNA polymerase reads the original DNA, so that's the one over here. That's the original DNA here. Or over here as well, that's the original DNA. And it inserts a complementary nucleotide. So over here what we have is here are the nucleotides, and the complementary ones are inserted. And then we have 2 copies. And down here, the polymerase, you see this green strand over here, the polymerase already has done that down here and over here on the other side as well. I'm going to show you now how this happens in more detail. So I'm going to erase again these lines here. And maybe it's a good idea to label. So that is our template DNA, our DNA template. So that's the original one that we want to copy. Whoop, what happened here? That's a weird thing. Okay. So that's the DNA template. Still, again, I have no idea. Okay, some problem with my pad over here. And over here we have the nucleotides. And these nucleotides are now used to actually make the new DNA. So how does this work? What we see over here is that the polymerase now, I don't know if you've seen this, let me go back one more. There is the polymerase which sits over here, basically reads the C, and it now will insert a G, the complementary one. Then the polymerase moves on one step, it reads that there is a T over here and it will insert an A. Then it will move on further, it reads that there is a C and it will take a G and insert it. Okay, so basically, and then all of these are, of course, are connected. And that is going to be our newly synthesized DNA strand. And you see that this newly synthesized DNA, I'm going to remove the polymerase again over here. This newly synthesized DNA contains over here, this is the original one, and this one is the new one. So, because that's what happens in semi-conservative replication. And now over here, what about on the other side over here? Well, of course, the same thing. You have a polymerase sitting over here, it reads the G, it inserts a C. And for the A over here, it reads the A and it will insert a T. So this is basically the polymerase acts, there's a polymerase on one side and also on the other side. And then, of course, I have again here a new DNA. And again, over here, this is the original DNA, the template DNA. And over here, we have the newly synthesized DNA. And then we have 2 DNA strands. And some students asked me already, well, when will the original DNA strands ever go back together again? Well, they will never do that. There is, they won't do that. There is no reason for them to go back together again. Over here in transcription, which I'm going to talk about later, this will go back together, but not in replication when we're copying the DNA. And when this is finished, then of course the helicase, which is still sitting over here, which I erased to make it a little bit, give it a better overview, the helicase will move on further and will separate these bases again over here, and the polymerase will take over and fill in the complementary bases. Other folks want to know, well, where do all of these nucleotides come from? Well, the body makes them, the cell makes them, or we eat them, because when we eat food, we are, of course, also eating DNA. And so all of these building blocks, of course, are provided by the cell, so that our cells can essentially copy the DNA. Yeah, so that was basically it. What I would generally recommend is I explained the whole thing now in the form of a diagram, but I would actually recommend that you summarize the whole thing also in a short paragraph or a text. And you can basically use this here as a basis. First of all, the helicase has to separate the DNA, and then the polymerase reads the original and inserts a complementary nucleotide and also makes a covalent bond. To make the new DNA, you need to make these covalent bonds between the bases here. And that is basically also something that DNA polymerase is responsible for. So that's it. That basically completes now DNA replication. And there is, of course, also a much more detailed version of DNA replication, but that is basically something that is relevant for higher level biology, and I'm not going to talk about this right now in this foil, but there's a separate video on that.